So today we're going to do biology ATP series related to all the questions that can come up your IGCSE biology paper 6. So we are basically talking about this topics, subtopics which relate to drawing and magnification, food tests and labeling, precautions and source of error, hazards, and drawing a graph, designing an experiment, methods, controls, and their uses, and sample question and a common independent variables, and like many more related questions. So, how do we actually solve this paper? I mean, paper 6. So we have like a lot of things to cover in this. So it'll be a short video, but yet a meaningful one. So first, starting off with drawings, how do you draw? The general rules for drawing is that you don't have to draw any details. Try to draw only outlines. First thing is when they ask for specific details, that's what you're looking for. Do not draw all the details like this is not gonna break out. So you have, for example, a cell like this, something like this. Okay. So there's a cell, then there is a nucleus here. So you're drawing the outline first, okay? Try to only outline, okay? Follow instructions and take care of labels that are asked in the question. If they tell you to label the nucleus, then only you're gonna label the nucleus. Do not draw arrows. Arrows not recommended. Arrows not recommended. Okay, then draw the drawing at least 50% larger. For example, this is what you want to replicate, so you'll draw it like very big. You get it like this big. Understand? So I hope you're understanding what I'm trying to say. So it is that big should be there. Don't use a ruler or compass to draw. Don't be like I'm gonna use a ruler, because you have to draw freehand drawing and no broken lines. Don't draw like this is not considered drawing, my dear. Okay. Read the question carefully and see if any specific segment is required. If there's any specific segment required, don't draw individual cells and don't draw surrounding box of the image. So you don't have to draw that this is the box and in this the image is lying or something like that. Magnification. When we talk about magnification, IGCSE paper 6, typically drawing the calculation questions are always followed. When you have the magnification formula, the question is typically asks you for 2 to 3 marks. It's pretty simple and you should know the formula. 1 millimeter is equals to 1,000 micrometer. So, magnification. When it comes to magnification, we divide the image size by the actual image. So magnification is basically I am. So I by A. So you gotta keep that in mind. Next is if the image size and magnification values are given and ask you to find out the actual size, you use the magnification of triangle formula. So you basically let me just show you. So if the image size and the magnification values are given, you ask yourself what you need to find. And this is a triangle by which you can just hide what you need to find and use that formula. I am. Okay, it's just like that. Next is food tests. These are the must food tests that you need to remember in order to be able to gain those marks in IGCSE. They talk about food test, which talks about starch. So for starch, um, you need to add drops of iodine solution. You need to observe color change. You need to check what is a positive result, blue black colored solution. Negative result is brown colored solution. Test for proteins. Add few drops of reagent. Observe color change. Positive results, mauve or purple colored solution. Negative results, blue colored solution. Test for reducing sugars. Add Benedict's solution and heat it. Observe color change. When reducing sugars are detected in a solution, the color change happens based on the increasing concentration of sugar. So first is blue, then it's green, then yellow, then orange, and red if it's with the highest concentration of the sugar solution. Positive test is a brick red, and negative test is a blue colored solution. Test for fat. Known as the emulsion test, crush the food sample in a mortar with ethanol. Take a sample in a test tube. Add drops of distilled water. Observe the milky white emulsion showing the presence of fat. Labeling. When it comes to labeling, you need to exactly point to the image and you need to use a ruler to draw the label line. When you're taking precautions, precautions can be many and you should know the precaution and how to avoid them. Take readings with the eye perpendicular to the scale. Very important. We can come out anywhere. Take readings from the bottom of the meniscus. Leave all test tubes in water bath to equilibrate. Hot test tube. Use tongs to hold the test tube. Stir the solution to distribute all the components evenly. While taking readings from measuring cylinder, place it on a flat surface. Wear a lab coat. Stand while performing experiment. Do not sit again. Don't expose an alcohol reagent to a flame directly. Heat it using water bath. Use burette or pipette instead of measuring cylinder because they are more accurate. For the synthesis experiment, use heat shield, a beaker of water placed between the plants and the lamp to prevent the temperature from changing due to changing light intensity. Next is heat using water bath, often used in Benedict's test for reducing sugar. And while using knife, you should use a solid surface to cut on and always cut away from fingers. Now, what type of errors can be caused? Very important. The same type of plant or fruit may not be used. Use the same species. So sometimes they don't like use the same species, which can cause a source of error in the entire investigation. Then measure error. So if you're measuring measurement error is caused if you're using graduated pipette. I mean, if you don't use a graduated pipette. Okay, water loss from tubes was not counted for. Cover the tube. So if you don't want the tubes to lose water, you should cover them so that evaporation doesn't take place. Reuse the syringe. So when you're reusing the syringe, it causes because if you reuse the syringe, it causes contamination. Reusing syringe, reusing measuring cylinder, reusing dropper, pipette, whatever that is, it can cause contamination, and a new syringe should be used each time. Subjective or quantifiable variable was measured. For example, measure a more quantifiable variable for accurate results. For example, if I say the color change is observed, then you will be like the color change depends person to person. That is why it's a subjective nature of color, and thus we use a colorimeter. No repeat. So repeat the experiment 3 times and find the average to remove anomalies. So why do we repeat the experiment? First is to make the investigation more reliable and to find the average to remove anomalies. Then we have when we talk about mass, volume, size, shape of pieces is not same. Sometimes the constant variables are not kept constant like mass, volume, size, shape. So which is also very important to keep constant. Next is uneven crushing, use of mortar and press pestle. So if you just crush with some kind of random pencil out in the desk and then say that okay it's crushed, but no, you should use proper equipment like mortar and pestle. Volume of each drop could vary. Measure the volume instead of number of drops. So sometimes it'll be like 5 drops are put. You shouldn't do that because you know that the volume of each drop could vary. Subjective nature, the use of steel range, measurement error, and not using the same species. Just take a screenshot of this because this is most common questions that come up in the examination. Temperature of the water may change to maintain the water temperature using thermostatically controlled water board. Heat may be lost through gap slit cork. Make sure lid perfectly fits the container. When a variable, example potato, is cut into smaller pieces, the smaller pieces are not measured to equal sides. This can be a source of error. The number of bubbles counted. Measure the volume of bubbles as the volume of each bubble may vary. Because if you say like, oh my goodness, I'll count the bubbles and see which has a higher rate of reaction, but the number of bubbles when you count, you know that the gas bubbles volume will vary from each other because some will be big bubbles, some will be small bubbles. That is the reason you need to measure the volume of gas instead of the bubble. Hazards. So, what are the hazards? Hazards are stuff that you should always look for when experiment planning. Hazards are not much, but they will get you the point. So, let's see. Cutting with knife may injure hand. Cut on the surface away from hand, and use gloves. H2O2 is corrosive. Use gloves, goggles while handling it. While exhaling through a tube of lime water, for example, there is a risk of lime water being swallowed. So, yeah, so be aware of that, okay? Iodine solution can stain clothes. Ensure gloves and safety goggles are worn. Next is drawing graphs. When you talk about drawing graphs, you use a sharp pencil, board axis labeled with independent variable on x-axis and dependent variable on the y-axis. Label should be in form of quantity slash units. So, for example, here is um, height in slash centimeter. Choose the appropriate scale. Use crosses or dot to mark the data point for scatter graphs, or mostly advised to use cross. Graph should cover at least half the grid. Draw a line of best fit that can fit through the most of the point as possible and join the points for line graph. Designing an experiment. Coming to the final part of this booklet, how do you design an experiment? The paper would consist of an example, and they would ask you to design an experiment based on the example provided, combining everything you have learned above. I'll show how to do an experiment planning question. General information. First, these are the general information. Mostly the marks located for these, the marks that are given are in a range of 6. So independent variable, if you tell 1 mark is going to be given details of the method, 1 mark. Constant variable, 2 marks because you need to give two constant variables. Safety precaution, 1 mark. Mostly they don't tell. Sometimes also dependent variable, another 1 mark. So the method, controls, and their uses. Method can be gas syringe delivery tube, measuring the volume of liquid. Ruler to measure the length of foam produced. Measure the rate of reaction by measuring the volume of product made. Cutting plant stem sections of every specified amount of time. Measuring the rate of water uptake every set amount of time. Counting the number of bubbles. Measure the rate of reaction by measuring the volume of gas produced. Measure the mass of potato stick before and after putting in a solution. Measuring the volume of water that is lost or gained per unit time. Measuring the height of a piece dough has risen. Measuring the rate of reaction in the piece dough by measuring its change in height. So, what can be controlled? Temperature. How do you control temperature? Thermostatically regulated water bath. pH of a solution. How do you control the pH? Use a buffer solution. Buffer solution are the solutions that will help you control the pH. Volume of solution. How do you control volume of solution? Using a measuring cylinder or gas syringe. Size of leads. How do you do that? Use a square grid. Time. Time can be measured using timer or stopwatch. Concentration, if you want same concentration of the named solution, same diameter of the same size or of testing. Additional tips are that stage 3 or more variations for independent variable. Example 5 concentration, 5 species, 5 acids. You can write either in paragraph or bulleted form. Don't spend more than 7 minutes on this question. Sample question. Use the mnemonic I don't care, so run away. I don't care. I don't care, so run away. IDCSRA. IV. So independent variable comes from I. Independent variable, you need to state the independent variable, describe how you're going to change it, include the name of the apparatus, state the value with units. Describe how you need to change it. Okay, very important. Okay, state the value. Then don't TV. State the independent variable, state the unit, state how you're going to measure it, how you're going to change this, how you're going to measure this. Okay. Then we have care, Stevie, which is state the three different control variables. Describe how you are going to control them. Okay. How you're going to control them. The first was how you're going to change them, you're going to measure them, and then you're going to control them. Then so run away. So safety. State a safety measure and how would you eliminate the risk. One, repeat the experiment 3 times. Away. Average. State that you would calculate an average if it's a numeric data. An athlete suggested the hypothesis. Drinking a greater volume of beetroot juice would increase the length of time the athletes are able to run. Plan an investigation to test that hypothesis. So first you follow this pattern. You write IV, the independent variable. So the independent variable here is going to be the volume of beetroot juice consumed. And that is what you're going to keep changing, right? How will you change? By using a measuring cylinder, right? And then we have the dependent variable, that is the length of time that athletes are able to run. So yeah, you're gonna calculate the time athletes are able to run. Then you're gonna control some variables like the concentration of beetroot juice, the maybe the species of beetroot juice, or the age of the athletes and the gender of the athletes, so on. Okay, then control variable. Yeah, we've done with control variable. How will you control them? Yeah, you don't have to tell that. You're just gonna pick a sample with the amount of, you know, age, gender, and other stuff. Safety. So when we talk about safety, when you are preparing beetroot, you have a chance of staining yourself. So use basically, what do we say? You use yeah, you're gonna use gloves and all this stuff. Then repeat the experiment 3 times and state what you and then repeat and then average. Okay. Prepare 20, 40, 60, 80, and 100 centimeter cube of beetroot juice. Consume the beetroot juice at 2 p.m. every day. Measure the length of time an athlete can run at their maximum speed. Concentration of beetroot juice, age, sex, the diet of athlete, same environmental conditions of exercise. Repeat with same individuals for many trials. Medical staff should be present. Marking scheme. At least 2 different volumes of beetroot juice. Details of when juice is consumed. Reference to fasting or any other wording. Details of the method to measure running time. Best breaks between repeat measurements. Named constant variables like concentration of beetroot juice. Correct details of consistency in the running. Age, stature, sex, fitness, diet, health of athletes. Same environmental conditions, right? For of exercise. Or many participants in the group repeat with same individuals for many trials. Safety precaution can be mentioned. Now, these are the common independent variable like temperature, how to changes, use thermostatically controlled water bath, a thermometer, measure the temperature, humidity, with or without plastic bag, light intensity, lamp, airflow, fan, and don't use a fan, carbon dioxide, different concentration of Na, NaHCO3, that's a source of carbon dioxide. So we are done with this IGCSE biology ETP. Watch this video again so that you are more clear and thorough with the understanding. Thanks for watching.